Immunology is an incredibly difficult aspect of physiology to comprehend. And one of the reasons why it's so difficult to really understand is because immunology is not linear. And what I mean by that is we don't have event A triggering event B, triggering event C, triggering event D, and it always happens in that order. Instead, we might understand what happens in event A, but we can't really understand the relationship between A and B. And why does A activate B sometimes, but B then comes back and activates A? And what about C? And what's this crosstalk between A, B, and C? And when is D activated? And I don't understand. And that's pretty much sums up how most people feel about immunology when they try to learn it the first couple times, or maybe the first 10 times. So in this lecture, I'm going to try to synthesize humoral immunity and cell-mediated immunity into one comprehensive but comprehensible story. And we're going to use a virus as our example because viruses always activate both humoral and cell-mediated immunity. If you are writing this out, I am going to be doing a giant flowchart to tell this comprehensive story. And if you want to copy it down, I strongly suggest getting a fresh 8.5 by 11 sheet of paper, turning it landscape style, and writing small, because we're going to put a lot of information in here. Much of the information that I cover will build upon the story that was told in lectures 3B and lectures 4, but I will be adding some additional information. And at the end, we'll take a look at memory cells and what function they serve in immunity. So let's get started. We are going to assume that we are getting infected with a virus. We have never been exposed to this virus. Or more specifically, we've never been exposed to the antigen that this virus is showing us. When you get infected by a virus, it is rarely a single virus that gets access to your body. It's usually a group of them, and we call this the minimum infectious dose. Right now for COVID, the minimum infectious dose is generally agreed upon to be a little over 100 to maybe 300 for the Omicron variants. So this means that when you get infected, you're not just getting exposed to a single virus, there's usually a group of them that come around the same time. So let's say that we get a whiff of these viruses up our nasal cavity. What happens to them? Well, some of them are going to get sucked up into the lymphatic system. So I'll put one over here under humoral. Some of them are going to actually attach and infect our cells. And some of them are going to be consumed by macrophages that were called over as part of the inflammatory response. So let's write this out. So now let's kind of address each one of these individually. The viruses that are phagocytosed by macrophages are going to induce that macrophage to digest them, pluck off the antigens, insert the antigens in MHC2, and then the macrophages are going to become antigen-presenting cells for that viral spike. So let's add that in. So what are the macrophages going to do next? They themselves are going to enter the lymphatic system. And who do they seek? They seek T cells, specifically helper T cells, with a complementary T cell receptor expressed. Let's look at the cell-mediated side. Viruses are going to infect the cells. And so what this is going to do is induce the infected cells to display the viral antigen on MHC1 in hopes that it attracts a cytotoxic T cell over to induce apoptosis. For reasons that I will not get into at this very moment, but will hopefully in a few minutes, that is not successful initially. The virus replicates, escapes the cell, replicates again, escapes the cell, infects new cells, replicates, etc. So we have this kind of unchecked viral replication at first. So the cytotoxic T cells are not yet able to induce apoptosis. And the why of that I'll discuss in a little bit. But let's put this in here. Okay, the infected cells that are displaying the antigen and MHC1, that is going to call over cytotoxic T cells. So now what we're doing is we are calling over cytotoxic T cells. Cytotoxic T cells are highly mobile and move around a lot in the blood especially. And so whenever you have a lot of anything in an MHC1, it tends to attract cytotoxic T cells to come and check it out and see if they have a complementary T cell receptor. Now, how long might this take? It could take a couple of days until you get the right cytotoxic T cell clone. And remember, meanwhile, these viruses are replicating and infecting more and more and more cells. So let us assume for a moment that it's 2 days later and the right cytotoxic T cell has finally engaged with the antigen and MHC1, and we've got the right clone, and it's bound to this infected cell. Based on the previous lecture, you know that the next step is to secrete perforin and then the apoptosis signal, but the cytotoxic T cell initially is not allowed to induce apoptosis. It has to be given permission by a helper T cell. So that cytotoxic T cell, even if it's the correct one, just sits there bound to the cell and hamstrung until it gets permission. And this is new information that I did not talk about last time. So let me go ahead and add this in, and then we'll take a look at, well, how do we get the helper T cells and what else is going on. Okay, our story is going to take a pause on that side of it. Let's come back over here. Let's come back over here to the humoral side of things. Viruses get sucked up into the lymphatic system. We know that the next step that we're looking for on the humoral side is for a naive B cell that has the complementary antibody to fish that virus out of the lymphatic system and become antigen presenting. However, in order for that to happen, that virus has to run into that rare B cell. And initially, that's unlikely to happen until the viral load in the body reaches a certain threshold. So some of these viruses over here that we're replicating also need to get sucked up into the lymphatic system until the viral load is sufficient enough that viruses basically flood the lymphatic system and they run into the right B cell. So now on the humoral side of things, we have a B cell that does have the correct antibody for this particular antigen that has become antigen presenting. But that's not enough to elicit clonal selection. It requires a helper T cell permission. Meanwhile, on the cell-mediated side, we have found the correct cytotoxic T cell, but it's not allowed to do its job until it gets helper T cell permission. So in this way, we are waiting for that macrophage to find and activate that complementary helper T cell. And now hopefully you're getting an appreciation for how all of these things have to happen to bring your acquired immune system on board. And since it's a constant game of trial and error, let me try this antibody against that particular antigen, let me try this T cell receptor against that antigen, and you continually try different receptors, different antibodies, it can take a while to get all of the players activated. And this is when that particular virus is having its way with you. In this case, I'm using COVID as an example. So you're getting sick and you're not mounting an effective response because you're waiting for all of these pieces to come online. So let's say the macrophage, which has gone from lymph node to lymph node to lymph node, finally finds its helper T cell. We know that the macrophage is going to bind to the helper T cell with cell-to-cell contact. And we know that when it binds, that antigen-presenting macrophage is going to secrete cytokines that will activate that helper T cell. This is a form of clonal selection. We have many T cell clones. We found the one with the complementary cutout shape. It's being activated. So when that antigen-presenting macrophage secretes cytokine permission to that helper T cell, that's the same as picking that helper T cell and saying you're the winner. You need to replicate and make a bunch of copies of yourself. Most of those helper T cells that are made from that mitosis and cell division are going to be active. But some are actually going to revert back to their naive state where they are inactive and pretend they've never seen their antigen. And the inactive ones are referred to as memory cells. So we don't just make memory B cells, we also make memory helper T cells. And we would make hundreds of copies. And this is like an army, an active army of helper T cells. But some of the daughter cells are going to be inactive. Inactive means they do not have the capacity to activate any other lymphocyte. They're not allowed to bind to anything and activate another cell. So these inactive ones kind of retain or regain their naive state, and we call them memory cells. Okay, so what happens now? The newly created army of active helper T cells are going to disperse throughout the blood, throughout the lymphatic system, going into tissues where there might be inflammation because that indicates something is wrong. Maybe there's some action going on there. And so we know that this army of helper T cells are looking for antigen-presenting B cells. So eventually we expect one of these helper T cells to go and find this guy. And we know it will be a perfect match. And when that helper T cell binds to that antigen-presenting B cell, the helper T cell is going to secrete cytokines and activate that B cell. So with clonal selection, that winning B cell is now going to produce a lot of daughter cells, most of which are going to differentiate into plasma cells, some of which are going to revert back to their original naive state where they advertise their antibodies. So we have now reached the end point of humoral immunity. Remember that antibodies are somewhat short-lived and plasma cells are especially short-lived. So antibodies, when they're secreted, have a half-life of maybe like 10 days in the body. And there are different types of antibodies. Remember, another way that you can name antibodies is called immunoglobulins. And these are abbreviated IG. And some of the IGs are IgA. We find these a lot in the respiratory system. IgM is kind of the typical one secreted into the bloodstream. IgD, IgE. So there's different antibodies, actually 5 different types, and they'll kind of differ in their function, but we're going to treat them as just one thing. So we're just looking at antibodies in general, and IgM is kind of your classic antibody. Okay, so we're secreting large numbers of antibodies that are going to be somewhat short-lived in the bloodstream, and plasma cells also die off rapidly after expending a huge amount of energy secreting huge numbers of these antibodies. In our story, we still have not resolved our cell-mediated immunity. So in our cell-mediated immunity, we kind of left off with the cytotoxic T cell binding to an infected cell. And again, based on the prior lecture, you should know that the next steps are to secrete perforin and then secrete the apoptosis signal. But I mentioned that we require helper T cell activation. Now, this is where I'm going to fudge a little bit, because in order for you to truly understand how the helper T cells might in fact bind to and activate a cytotoxic T cell requires a more comprehensive study of T cell receptor design. And I hinted at it a little bit when I talked about CD4 and CD8, but again, I'm going to pick my battles, and that's really going to be beyond the scope of what I want to talk about. I will say this: helper T cells and cytotoxic T cells come in a pair. When the thymus gland matures a T cell, it goes through a negative addition. And what that means is that that T cell cannot bind to anything that you make. If it does bind to something that is self, that T cell is removed from consideration and it's destroyed. But as long as that T cell does not bind to anything that you make, it potentially is allowed out into the circulatory system or the lymphatic system. But before that T cell is released, it splits into two, one of which becomes a helper T cell, and the other of which becomes a cytotoxic T cell. So what that tells us is that for every specific T cell receptor that you find in a helper T cell, there's a matching T cell receptor shape on a cytotoxic T cell. Now again, for my class, that's a little bit beyond what I need you to know. I did just want you to know that they do come in twinsies. So what we're waiting for is one of these helper T cells to come over and basically identify this cytotoxic T cell as its twin and secrete cytokines to activate it. And how that works, again, is something I'm just glossing over. But you do need to know that initially, helper T cell permission is required for the cytotoxic T cell to be active. So now that cytotoxic T cell has been given permission and it's ready to do its job. It's of course going to kill the infected cell, but the other thing it's going to do is replicate and create a bunch of identical daughter cells so you can get an army of these cytotoxic T cells. You do need an army because at this point you are infected and really infected, and you probably have millions of cells that need to be identified and killed. So we need an army of these too. Okay, this is a pretty extensive flowchart. I'll kind of zoom out a little bit here. You can see just how extensive this flowchart is as we learn about the comprehensive story of acquired immunity. And if you were able to follow all of that, you should be very proud of yourself because this is a lot of information and there's a lot of moving parts. And again, it's not always a linear story, which makes it especially difficult to understand. I'm going to address memory cells in just a moment, but before I do, I wonder if you've noticed that I didn't use the word memory cells with cytotoxic T cells. In other words, I did talk about creating a bunch of active cytotoxic T cells, but I didn't refer to it as clonal selection, and I didn't talk about memory in active forms. Cytotoxic T cells function a little bit differently than B cells and helper T cells. Whereas B cells and helper T cells do produce daughter cells that are truly inactive and truly revert back to a naive state, cytotoxic T cells do produce kind of an inactive form but they can become reactivated simply by binding to the antigen in MHC1. That is to say, they can reactivate themselves. Upon the very first time you're exposed to a particular antigen, cytotoxic T cells require permission from helper T cells. But once that permission has been granted, the daughter cells can be reactivated themselves for life. Subsequent exposures to the same virus, so if you happen to be exposed to COVID again, the cytotoxic T cells that were still left behind from the first exposure, if they bind to an infected cell, will kill the infected cell. They don't just sit there waiting in purgatory for a helper T cell to come over and give it permission. They'll just kill it, and then they'll reactivate themselves. So that helper T cell permission is only required once for each cytotoxic T cell clone, which I think is really interesting. So it's kind of like a clonal selection, but it doesn't create memory cells in the same way that helper T cells and B cells do. Okay, so feel free to go back and rewatch this as needed to really get the story to stick in your head. But at this point, we're going to move on and we're going to talk about these memory cells and what the importance of these memory cells are, or even having additional cytotoxic T cells, the army, that might be reactivated at any time. So let me get right to the punchline. Memory cells are what confer immunity. So it's generally understood that when you get exposed to a particular pathogen or antigen once, and you get sick from it, if you get exposed again, you should not be getting sick again. That's what immunity is. So it's a very black and white way of thinking about it, and the reality is much more 50 shades of gray. So what I mean to say is that we can have different degrees of immunity. If you're fully immune to something, I could throw 10,000 viruses up your nose and you would never be symptomatic and you would not be a risk for transmitting it to somebody else. That kind of immunity is actually, I would say, more rare than the type of immunity we see most often in the community, which is kind of a partial or a weak immunity, which means that if I throw 10,000 viruses up your nose and you're partially immune or you have a weak immunity or a waning immunity, then they might start to replicate, but you squash the infection very early. So if you are symptomatic, the symptoms are more mild and or more short-lived. And I would say that probably is more common than having complete and full immunity to a particular disease. Now, there's a couple different ways to get memory cells. One, of course, is to get really sick, but the other is vaccinations. So the goal of vaccinations and immunizations is to generate memory cells to a particular antigen. Vaccinations and vaccine design is going to be covered in the next lecture. For now, let's assume that we get these memory cells from actually getting infected. So memory cells are going to make you immune, and this is because it's a game of time. The reason why you get sick in the first place is because you do not activate acquired immunity quickly. And why is that? Assume that we have a helper T cell in this lymph node, and we have the naive B cell that we want in this lymph node over here, and we have a naive cytotoxic T cell somewhere in the blood over here. In order to activate acquired immunity against a virus, you have to find this single needle in a haystack, and it's a really big haystack. But first, you have to find and activate this needle in a really big haystack. Imagine if we were to go to a very large parking lot, like at a stadium, that had 10,000 different cars in it, and I give you a single car key and there's no identifying information about that car key. You don't know if it's a Honda, you don't know if it's a Toyota, you just know that it's not a fob. And so you actually have to try the key out in the lock, and you go around from car to car to car to car to car trying to open the doors. How long is it going to take you until you find the right car if there's 10,000 cars? It might take you 5 minutes if you get super lucky, but you're probably looking at many, many hours. And in the same way, when you have never been exposed to a particular antigen before, you don't have very many copies of the native or naive B cells and T cells. And so you have to find them. And it's like finding that one car out of 10,000 or the single needle in a very large haystack. And you don't just do this once, you do it several times, because you have to find and activate the right helper T cell, and find and activate the right B cell, and find and activate the right cytotoxic T cell. So it's a pretty long process. And it can take 5 days until you start secreting antibodies. It can take 7 days until those antibodies are measurable in your blood. Okay, so what has that virus been doing with you in the meantime? Infecting your cells? Perhaps extensively? And this is what's happened in COVID. So when people had never been exposed to COVID before, like earlier in the pandemic, then they would get exposed and COVID would infect a lot of their cells very aggressively while the body tried to fumble around and find the right naive helper T cell and the right naive B cell and the right cytotoxic T cell and bring them all online. And by the time you've activated all of these, people's entire lung tissues would be infected. So we have not yet learned about the respiratory system, but you're going to see that the lungs are made of simple squamous epithelial. That means a single layer of cells. And if all of those cells, or a good part of those cells, are infected, and now you've got your cytotoxic T cells are now going to go and destroy them, you're literally destroying your own lungs. And that is acute respiratory distress. And people often died of that. So, if you have been exposed to COVID once, maybe you've got COVID and recovered, or you were vaccinated for COVID, the idea is that you've generated memory cells. And when you've generated memory cells, you put more needles in the haystack. Those memory cells that you can now see here get redistributed throughout the body. So, instead of having a lot of difficulty finding the right lymph node or the right corner of your bloodstream, ideally you would have enough memory cells that basically every lymph node would have a complementary helper T cell, a complementary B cell, and a complementary cytotoxic T cell. And so where does this leave you? This leaves you with a macrophage. Who knows where that macrophage might be coming from, but that original antigen-presenting macrophage is going to be able to find a helper T cell in the very first lymph node it goes into and activate it. And so what that's going to do, of course, is create an army of these helper T cells that are immediately going to go out and try to find a B cell. And meanwhile, that virus, which may have come from the same, general area of the macrophage, is coming in, and the first lymph node it runs into, guess what it runs into? The matching B cell. And that matching B cell becomes antigen presenting. So you can see it will not take very long before antibodies are produced in large quantities. And also, if there happens to be any infected cells, one of these cytotoxic T cells is going to go find it, become reactivated, and destroy it before it has the chance to allow for a large number of viruses to start circulating around the body. So in this way, you are able to find the complementary B cell, the complementary helper T cell, and reactivate the cytotoxic T cells within a matter of hours, rather than a matter of days. And you squash the viral infection before it really gets a chance to get a toehold in your body and create symptoms. So this is how memory cells confer immunity, and it's really awesome. So the original design of the body is one where we diversify our portfolio and we create a lot of different B cell clones and a lot of different T cell clones. But you do not know which T cells or B cells will ever actually be useful, because you're creating those antibodies somewhat randomly and you're creating those T cell shapes somewhat randomly. But when you do find a B cell and a T cell that is useful in fighting an infection, your body says, hey, since I've seen this once, the likelihood that I'm going to see it again is high, so I better put more eggs in that basket and make sure that I have enough T cells and B cells of that particular clone so that the next time that pathogen walks into my body, I'm prepared. So in this way, the immune system cleverly adapts by changing the proportions of various B and T lymphocytes depending on what you've been exposed to. So let's quickly do kind of a graph that shows how the antibody levels will respond in your blood based on the first exposure to an antigen, meaning you've never seen it before, and after you've generated memory cells. So let's assume that day 0 is when you actually get infected. So the first time that you get exposed to an antigen, it should follow a trajectory like this. No antibodies, no antibodies, no antibodies, because remember, we're trying to find the right T cell and activate it, and then the right T cell has to go and activate a B cell, but the B cell has to have seen its antigen. And the only way that's going to happen is if the antigen's very numerous. So that might take a little while. And then finally we get some antibodies that come out, and then they peak a few days later and then slowly decline over time after that. The second time you see that pathogen, we call this secondary exposure, you're going to have a response that looks like this. So notice that not only do you bring out your antibodies much more rapidly, but you have a lot more of them too. The rapid response is because we are no longer looking for that single needle in a haystack. There's now hundreds of needles. The first lymph node you go into, you're going to start activating stuff. And with any luck, you're going to bring out your antibodies in a matter of hours. But what's interesting is also that we get a lot more antibodies the second time around. That is because the macrophage isn't going to stop there. It's then going to go into the next lymph node and do the same thing. And so these are going to activate all of these guys, and we'll get some antibodies from here. And then it's going to walk into the next lymph node and do the same thing. So we're going to be activating multiple T cells and multiple B cells, and therefore getting a lot more antibodies out, which is only going to reinforce the superiority of your immune system in squashing its exposure to this pathogen. Okay, we covered a lot of information here, but hopefully you're able to follow it. And my final lecture on immunology will look at vaccine design and how vaccine design can influence the amount of memory cells you generate. I'll see you then.